Welcome to week 2 in the Math and Science of COVID. This week we will discuss the last year and a half of the COVID-19 pandemic. Although we won't be able to discuss everything that has happened over the last year and a half, we will discuss important events, milestones, and discoveries that are specific to the math and science of COVID. This might be a little dry because I'm going through a timeline, but stick with me, the course is going to get more exciting. See you soon. "2020 was a year we will never forget. Lives were lost and lives were changed forever. Although 2020 will rightfully be identified as a year of horrific tragedy, it was also a year of significant scientific innovation. In this video, I will present a review of many, but not all, key events associated with the COVID-19 pandemic. I will not attempt to list all of the significant events that occurred, but rather focus on the scientific and clinical events pertinent to this basic course on the math and science of COVID-19. My hope is that this will serve as a springboard to elaborate on these topics in our other video modules. Many thanks to the healthcare and research communities who rose to meet the challenge of COVID-19. The information in this video has been compiled from the following sources: the World Health Organization, the Center for Disease Control, and Johns Hopkins University. On December 31st, 2019, the WHO reacted to a media announcement from the Wuhan, China Municipal Health Commission. The statement from the commission reported 44 cases of what was identified as viral pneumonia. By January 5th, 2020, the WHO notified all member states about the outbreak of a viral pneumonia of an unknown cause and advised precautions to reduce the risk of infection. As part of this announcement, the WHO reported the number of infections and the clinical status of those infected. Early announcements indicated that the source of the outbreak was an open-air seafood market in Wuhan, China. However, there has not been sufficient evidence to support that statement. It is most likely that the market was a superspreader event that resulted in human-to-human transmission. On January 7th, Chinese authorities identified the pathogen as a new coronavirus. By January 12th, the genetic sequence of what was then referred to as 2019 nCoV was published, which was a day after China reported the first death from this novel coronavirus. By January 13th, Thailand's Ministry of Public Health reported the very first case outside of China. On January 10th, a first draft of the viral genome responsible for COVID-19 was published. On the 14th that same month, during a press briefing, the WHO reported that Chinese authorities didn't have clear evidence of human-to-human transmission. Less than a week later, human-to-human transmission was confirmed, but the degree to which had not yet been determined. As of January 23, 2020, all but 10 of the 581 global cases were in China. One of those 10 cases outside of China was confirmed to be in Washington State, here in the United States. The next day, French officials confirmed the first 3 cases in Europe. By January 30th, there were also confirmed cases in the Eastern Mediterranean region. By this time, there were 17 confirmed mortalities, all within the People's Republic of China. On February 11th, the International Committee on Taxonomy of Viruses named the novel coronavirus, currently known as 2019 nCoV, to Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. That same day, the WHO began referring to the syndrome as COVID-19. Within days after that, scientists were able to pinpoint the receptor that the virus used to gain entry into human cells. On February 24, 2020, the WHO held a press conference urging nations to implement their highest level of national response management to try and contain the spread of the disease. By the end of February, the COVID-19 case count was already approximately 10 times that of the 2002-2003 SARS outbreak. On March 11th, the WHO declared COVID-19 a pandemic. Also, during March of 2020, scientists were able to isolate the virus from patients who had contracted it. As such, they were able to isolate the specific antibodies against the SARS-CoV-2 virus from patients. This led to the development of commercial tests that would allow the identification of the presence of those antibodies. This flood of information enabled researchers at the NIH's Vaccine Research Center, in collaboration with pharmaceutical company Moderna, to begin a clinical study of what would become the first FDA-approved vaccine for COVID-19 in January of 2021. It is important to realize that this literally began as early as 2 months after the initial draft publication of the SARS-CoV-2 genome. As we moved into April of 2020, clinicians began to report that their patients were experiencing a loss of smell and/or taste. Italian researchers followed up on these clinical reports and conducted a study on these neurological symptoms. They found that 64% of patients with mild COVID symptoms in their sample were experiencing a loss of taste or smell. Additional neurological symptoms emerged in patients with other comorbidities. In May 2020, the Chinese company CanSino Biologics announced clinical trials of an adenovirus-type vaccine. We're going to talk a lot about adenoviruses in a future video. This is a different type of vaccine than NIH Moderna vaccine, which is an mRNA-type vaccine. And like I said, we'll unpack these types of vaccines further in an upcoming video that I've entitled Viruses, Variants, and Vaccines. Following up on their phase 1 clinical trial, Moderna announced that its mRNA vaccine was both safe and effective. Also, Pfizer and BioNTech announced human phase 1 trials of 4 RNA-based vaccines against COVID-19. Also in May, investigators were beginning to study SARS-CoV-2 DNA vaccines that were specific to 6 different spike protein variants. We'll talk a lot about the spike protein and variants in our upcoming video modules. On May 14th, investigators published research that demonstrated that 30 to 50% of people tested have a form of pre-existing immunity to SARS-CoV-2. This type of immunity is called CD4 positive T-cell mediated immunity. Now, I know that's a mouthful, but we'll unpack that in our basic immunology module. Unfortunately, these findings were overemphasized by some media outlets. They suggested these findings were evidence that humans were already protected against COVID-19. The researchers never made that assertion. It's thought that this pre-existing immunity may have come from other seasonal forms of human coronaviruses. So in June 2020, we saw a surge in therapeutic drug studies for COVID-19. Dexamethasone, which is an anti-inflammatory agent, remdesivir, hydroxychloroquine, and other agents were subjects of randomized clinical trials. Dexamethasone and remdesivir were the most encouraging, whereas the popular hydroxychloroquine treatment showed no significant pre- or post-prophylaxis in COVID-19 patients. Broadly, these studies helped clinicians formalize COVID-19 disease interventions that were stage-specific. The summer months of 2020 produced several publications that bolstered our understanding of how antibodies protect us from COVID-19 infections and reinfections. Also in June, Regeneron Pharmaceuticals published data about their monoclonal antibody therapy and demonstrating its efficacy. Their study also helped us begin to understand emerging natural variants, and we'll talk a lot about those variants in our vaccines and variants module. Research published in July 2020 measured specific serum cytokines, which are a type of cell-to-cell signaling protein in hospitalized COVID patients. These investigators were able to determine that high levels of certain cytokines during hospitalization were actually good predictors of patient mortality. This study was followed by several others that furthered our understanding of the body's immune response to COVID-19. Moving into the autumn of 2020, scientists began to find early evidence of autoantibodies playing a role in slowing the recovery from COVID-19 or even contributing to long COVID. Autoantibodies are like the antibodies found in autoimmune disorders. These are basically rogue antibodies that turn on the individual producing them. We all have them, but some people may be genetically predisposed to produce more. More research is really needed to fully understand the role of these autoantibodies in COVID-19. Also, very interesting research emerged about the role of interferons in COVID-19 between the months of September and October 2019. Investigators found that type 1 and type 3 interferons, which are a type of cytokine that controls or even inhibits viral replication in our cells, and that research discovered that type 1 and type 3 interferons are very important. Specifically, if there's some form of mutation that inhibits the induction of these signaling proteins, or if patients produce antibodies against them, yes, that would be considered an autoantibody, those patients are at greater risk of life-threatening COVID-19. As we moved into November and December of 2020, we began to see a rise of the viral variants. In November, the virus began to transmit from humans into the mink that they were handling. Mink being the cute little animals that some people used to make coats out of. The virus was adapting to the mink by introducing a specific mutation in the binding site of the spike protein. We'll talk a lot about mutation. We'll talk about binding sites and the infamous spike protein in our upcoming virology vaccines and variants modules. But to continue, this mutation caused scientists to take note of the rise of SARS-CoV-2 variants. This escalated in December when the UK variant and the South African variant were identified and determined to be more infectious than the virus that originated in Wuhan, China. And again, we're going to cover variants here in a little while. The good news that happened in November of 2020 included two major announcements related to the COVID-19 vaccine. On November 9th, Pfizer and BioNTech announced a 90% efficacy rate for their mRNA vaccine candidate. The following week, Moderna reported a 94.5% efficacy rate for their mRNA vaccine candidate. In early December, both Pfizer and Moderna announced final efficacy data of 95% and 94% respectively. Both Pfizer and Moderna vaccines were authorized for emergency use in the US and the United Kingdom at that same time. Also in December, AstraZeneca announced early efficacy data for their vaccine candidate. This viral vectored vaccine, which we'll talk about later, was reported to be just over 70% effective. Other viral vectored vaccines from across the globe were announced as well during December. And again, we're going to talk about the difference between mRNA vaccines and viral vectored vaccines in our viruses, vaccines, and variants module. Now, although the COVID-19 vaccines were developed in an unprecedented 11-month time frame since the beginning of the pandemic, where COVID-19 moniker was assigned, close to 100 million people worldwide were infected, and 2 million people had died from COVID-19. As we moved into 2021, we faced challenges of getting these vaccines to our global population. Shifting into 2021, the WHO hosted a forum to discuss the status of research on variants and shine a light on existing gaps in the research. Now, the key points of discussion at this forum included mathematical modeling, animal models, diagnostic tools, vaccines, and therapeutics. Shortly after that WHO forum, about a dozen scientists from the International Virus Origins Mission arrived in Wuhan, China to work toward a better understanding of the origins of the SARS-CoV-2 virus that led to the COVID-19 pandemic. On January 26th, an agreement was signed to dramatically improve equitable and fast access to the COVID vaccines worldwide. The organization responsible for this initiative was known as COVAX. The goal of COVAX is to provide access to 2 billion with a B, vaccine doses by the end of 2021. In late January 2021, the CDC reported the emerging variants that are circulating across the globe. At this point, the most notable variants were identified as the UK variant, the South African variant, and the Brazilian variant. Also, the WHO issued recommendations on the use of the Moderna mRNA vaccine, which included recipient, priority, and dosage. Also, the WHO issued a statement of confidence of the Moderna vaccine's efficacy against the B117 and 501Y V2 variants. We're going to talk more about variants here in a little bit. On February 15th, the WHO announced its approval of the AstraZeneca vaccine for emergency use. On March 12th, 2021, the WHO announced its approval for the J&J or Johnson & Johnson vaccine for emergency use and gave its recommendations on how to use this vaccine 5 days later. The number of Americans infected by COVID-19 variants of concern, which again are UK, South Africa, and Brazil, is just over 20,000 patients at this point. And we're going to unpack the variants and what they're called, what they were called, and what they are called now in a future video. On April 13th, 2021, the CDC recommended a pause, in other words, a brief stop, on the Johnson & Johnson vaccine out of a, quote, abundance of caution, end quote. And here's why. Out of the 6.8 million doses that had been administered, there were 6 recipients of the vaccines who experienced a severe type of blood clot. 2 days later, the global mortality rate of COVID-19 surpassed 3 million individuals. On April 27th, the CDC announced that vaccinated individuals were safe to resume their pre-pandemic outdoor activities and even some indoor activities without a face covering. In mid-May, the CDC's guidance on face coverings loosened even further. Now, to better monitor emerging COVID variants, the WHO elected to add a Greek letter naming convention on May 31st. So that's what we talked about earlier with all the B.1.1.7. You know, we're going to make it a little simpler now to follow these different variants. So on June 3rd, there was good news and bad news. On June 3rd, the number of active cases of COVID-19 dropped to its lowest level since March 2020. However, shortly after that, the United States reached the tragic milestone of 600,000 COVID-19 deaths. 2 weeks later, the WHO announced the global COVID-19 death toll had just exceeded 4 million people. In early July 2021, the SARS-CoV-2 Interagency Group, or SIG, in the U.S. created a classification system to better facilitate surveillance of variants. These classifications are variant of interest, variant of concern, and variant of high consequence. While there aren't any variants considered of high consequence, there are 4 variants that are classified as variants of concern. Those are alpha, beta, delta, and gamma. We'll spend more time differentiating these variants in our upcoming viruses, variants, and vaccines module.